said, a couple people said that they couldn't be here, but I said,
You want to make me co-host, Bobby, so I can let folks in if you're screen sharing?
yeah, let's do that. Okay. I'm gonna make you co-host. Yeah. All right, and then I'm going to screen share, but I thought what we'd try to do is, I don't think I'll go over every part of that matching exercise, but I'll start out by asking questions if people have any particular problems with it. I'll say one thing that I noticed, that plant anatomy, well, let me show you something that Donna brought up one time, made me think about it. Let's see, plant anatomy. Oh, goodness, here it is. Oh, I have to share a screen. Geez. All right. Let me share the screen. Yeah, excuse me. I think I got the flu I was telling. I feel like I do, but I don't have a sore throat, so it's probably not COVID. I'm sneezing and just all kind of, what the heck is this? Didn't ask for that. Jeez. Let me see if I can X out of that without losing us. Okay, well, okay, let me get out of that. All right, now, the question comes up, and this was in the slideshow about the second one. Why are we talking plant anatomy? And the biggest thing that people probably want to be in a society for is learn how to identify plants, and classifying is part of that too. Well, technically systematics or biosystematics, which is the more modern concept of what used to be called plant taxonomy, involves all of the biology, and anatomy and morphology are part of it. And so that's why this diagram is what it is. And I wanted to point that out. Now, let's see, can you see that? Can you see these books? All right, all right, in terms of helpers for the matching is what I'm getting at. This one here that I recommended in the beginning, Plant Identification Terminology by Harris and Harris. Can everybody see that? Okay, that was almost worthless when it came to plant anatomy. So, but this other book that I know Donna has, and I had mentioned Dictionary and Botany, is a more comprehensive book. The other book's more about plant identification. Plant anatomy is a subtle part of plant identification. It's not an overt part of it, but Dictionary of Botany has, let me give you an example, two terms that both relate to the root, anatomy of the root and some stems, particularly rhizomes. Endodermis and pericycle. Those are terms on the matching. And those two terms weren't in weren't in Harris and Harris, but they were in Dictionary of Botany. And they are all, those terms are also in, let me go to something here. Let's see. This was one of the handouts I gave you that was useful. It's a 1993 vintage thing that I put together, and those that handout was a particularly good handout. There's endodermis, and then pericycle is in there too. But if you use the tools I gave you, which when things are electronic, you tend to lose sight of them. So let me, so that sometimes is a problem with that. Some other handouts, plant anatomy diagrams. This one was particularly useful if you, let me rotate that. I thought I was. Rotate to you. Counterclockwise. That's a hand done one, but it's still kind of good. And this one is especially good. That's an ancient one, but it's just loaded with information and particularly useful diagram. But so those were some of the tools that you had at your disposal. This was another one that you didn't really get into much, but if when the cell divides in this direction, we call that periclinal division, and the one that is parallel to the surface, whereas an anticlinal division, it's like has to do with increases in girth, and that's what's going on there. So understanding how cells divide involves thinking 3D, and that's where good microscope work comes in. Let's see. Now, in terms of handouts, I just sent out the invite for the upcoming session. And this was an attachment that I didn't talk about. And this was an attachment that I didn't talk about. What's the other one? Wait a minute, that pine. Well, there were two attachments, and they were both pine cones sections of young pine cones when the cone is only about an inch long. And this is where the seed is going to be, right in this region here. I don't know if y'all can you see my pointer?
Yes.
Yeah, I wonder if I'm better off with this annotate thing. Let me see if I can get to that. Spotlight, I think it is. It might be a little, yeah, there it goes. Now if I can just get rid of that. But anyway, that's a little more obvious in the pointer. But anyway, those were part of the upload for the upcoming session. And which is more like a focus on morphology for reproductive structures like cones, of which they were in flowers. And so it's still carrying on morphology, but more focusing on the morphology and somewhat anatomy of reproductive structures like the cones and their analogs in the angiosperms, the flowers. All right, let's see. Let me get rid of that. Let's see if there's something else in here. Did anybody else come in, Steve?
So far, nobody else.
All right, all right. Oh, by the way, did y'all sort of parallel to this, that in the recent edition of the newsletter, I got the first installment out of my series on woody plant identification problems. And I've got good response from people on that. So I'm hoping y'all are noticing it too. This right here is, let me go down to ST. Uh-oh. It's not going to let me do the stipules. Oh boy. I thought I could do a search. I wonder if I still can. Okay, good. STIP. Wonder what wouldn't before. Okay, now understanding stipules. What I'm doing is still bridging the gap between anatomy and taxonomy, trying to show you how it's useful. This article here, well, this whole article is too long for one newsletter, so it's going to be in 2 or 3 installments. But this part about stipules is related. So stipules, when it comes to identification, or I think it was actually, yeah, stipules were on the matching, about the third from the number 33 was stipules. And let me read the definition while it's, I'm actually defined them on that sheet, I think, but gosh. Stipules are usually a leaf-like structure at the base of some leaf petioles, often or usually comes in pairs. And a good example of that is should be right in here. I saved it. This is an illustration that's going to be in the newsletter upcoming. That stipules on wild black cherry or wild cherry, and there's stipular pair there the stipules fall off though, and that's the norm for them to fall off like that. This is what they look like on holly. There's one of them. The other one is on the other side. There's a bud, young bud there in holly. Hollies tend to have little black or maroon stipules like that. And you really have to be looking at new growth to see them. And then the stipule falls off. What do? Well, then you start looking for the scars. And even though this is not a woody plant, it's tick trefoil. It's the things that get on your pants that aren't stick tights. But anyway, there's one stipular scar there that's that you see when the stipules fall off. So now, with that in mind, let's look at this statement. Leaf scar, okay, stipules are related to the anatomy of the node. As stated previously, and by the way, if you can master stipules, you can really exponentially get ahead in identifying some plants. The node is a place on a stem where leaves occur. That's what a node is defined as. Leaf scars are also at the nodes. There are two rules for plant anatomy that are useful here. Plant anatomy that pertains to plant identification. Most woody plants that have 3 vascular bundles in their leaf scars have stipules. Species of Nyssa are an exception. Now, I'll show you the exception. If I can find it. I had it here. Oh, I did have it here. Oh, here it is. All right. Okay. There is 3 veins or bundles in the leaf scar of a swamp tupelo, but there's no stipule over here or even a scar, or there's not one here. So that's an exception to this rule, but to the number 1 rule. That if something has 3 bundle scars like that, you'd expect it to have stipules. Now, another exception, the second rule is that in almost all woody plants that have 1 bundle in the leaf scar, stipules are absent. Species of holly are the exception. Now, if you go back and get that holly again. If we tore that leaf off, we would see only 1 bundle vein in there. And so having stipules is an exception. So holly is an exception and Nyssa is an exception to the rule. Normally when you have 1 bundle scar, you have no stipules. Normally when you have 3 bundles scars in the leaf scar, you have a pair of stipules. And that goes back to some research back as early as 1914. So anyway, so I wanted to point that out. And I think that pretty well addresses that takes care of that. The whole point of that is I wanted to show you that plant anatomy is useful for identification. It's also useful for classification. Classification, you start getting involving in higher levels like family concepts and family recognition. Let's see. Let's go back and point out something that we had a moment ago. Let's see. I think these plant anatomy diagrams will be part of it. Let's see. Okay. Okay. All right. What do you call this structure right here? That's a vascular bundle. Okay. And a vascular bundle is basically a vein, and that's one in the stem. Now, normally, all right, if I ask you, would the phloem be on the outside out here or the inside? And the answer is the phloem is on the outside and xylem is toward the inside. And that's the norm. But that's the norm in stems. But in a family like cucumber is, the family is held together by, I think just about everything in that family has what they call bicollateral bundles, which means phloem is on the outside and phloem is on the inside. So there's phloem, xylem, and phloem. So that's an example of how anatomy can be useful in classification besides just ordinary identification. So, but that's that's a pretty high level thinking skill when you start learning families and studying anatomy and putting that together. But those are cases I wanted to point out. All right, now nothing. Let me. I think I can stop the screen sharing and come back. And nobody else has come in, so we still have 9.
We had two more come in, Bobby.
Oh,
while you were in
okay.
screen sharing.
Okay, well, good deal. All right. Anyway, so I found I put the matching together myself, and so I decided I should be able to take the exam myself and do okay. Well, I did, and I'd be pretty bad if I hadn't, because but I did it without looking anywhere. But I'll tell you this: there were ones that were hard that I had to do by the old-fashioned process of elimination. I had, let's see, primary meristem number 11 I thought that was hard. I found vascular bundle, which I just pointed out at 25, shouldn't have been that hard, but it turned out to be for some reason. A strand, the key word is strand. That's what I just showed you. A strand, basically a vein, a strand of vascular tissue composed of xylem and phloem and possibly procambium, if still present. Procambium is a meristematic tissue that forms xylem and phloem, primary xylem and primary phloem. But so that was, I don't know why it was hard, but because it's just, I think I didn't focus on the word strand, which is the leading one, practically the leading word there. Anyway, the ones I'm pointing out now, the 4 that I'm pointing out now, I saved to the last. For some reason, they seem more difficult. Primary tissue at number 9, cells or tissues derived from the primary apical meristem of the root and stem. That was one of the latter ones that I did toward the end. And this one I understand, ground tissue, the very expression ground tissue sounds ambiguous or vague. But that's number 3, and that's a tissue making up most of the primary tissue of plants. The pith is full of it, so is the cortex. Pith and cortex, by the way, are regions, not tissues. But anyway, that's all I'm going to say about that matching exercise, unless anybody else had any problems or questions about it. Did anybody? Somebody said, I don't remember exactly who, it doesn't really matter, for some reason missed the exercise in terms of an attachment. And I wound up sending them the answer sheet before they saw it, but that's okay. If they were played the game right, they could still do it. Just don't look at the answers before you do it. But as I was saying, for those that join later, the Harris and Harris book that I talked about in the first lecture was worthless when it came to anatomy, like pericycline and endodermis weren't there. And those are not unique to roots, but are almost unique to roots and tissues that are. Now, if I ask you what the pericycle is for in the roots, the biggest function you should remember is that pericycles give rise to the tissue gives rise to lateral roots. And then, then what gives rise to lateral branches? And that comes straight off the apical meristem in lateral buds. They call them bud primordia. Young baby buds are called bud primordia. So anyway, so the pericycle's function in the roots that doesn't exist in the stem for that purpose is to form lateral roots, branch roots. Now, root hairs weren't talked about a lot, but root hairs are just one-celled entity. So the epidermis, and they are. God, that picture. Steve's got that guy. Anyway, I forget who he's supposed to be, Steve. But anyway,
well, that's Igor. Marty Feldman.
Yes. Marty, what's his name?
Marty Feldman.
Yes. I couldn't think of his name. Anyway, I'd rather look at your face than him, though. But he's distracting. But anyway, okay, anybody have any problems or questions that I didn't address? All right. Let's see if there's anything else I can think of that needs to be said. Well, without reviewing the whole thing, I can't think of anything. But if nobody's got any problems or questions, we'll disband. Amy, you got anything? Guess that's a no. But now I'm surprised I still got like 27 people in this, and the beauty of it is I guess that you can do it on your own time. And there was quite a lag in this last section because of my travels, but that was probably a good thing, because this was probably the most difficult, one of the most difficult topics that there is, and largely because plant anatomy, there's probably a handful of schools in the United States you can still take a course in plant anatomy, but I've actually taught it myself, and it's a chore, and you got to have a good basic botany class first, because otherwise you're at a disadvantage. In fact, one of the advantages to this class is a good cell biology class, a good basic biology class. And by the way, I don't know if you remembered or not, but I found that the basic courses in every discipline, I don't care if it was sociology, psychology, or biology, the basic courses are the hardest, because you cover a lot of material and don't spend much time on each subtopic. You know, embryology, botany, ecology, zoology, invertebrate zoology. You don't spend much time on the genetics. You just kind of trip the light fantastic in a basic course, and you don't. And when you take an advanced course, you're repeating some of what you danced across in a basic course. And the basic course, when I got about time I got through teaching, the basic biology book was so heavy you needed a wheelbarrow to take it around. But anyway, well, that's all I got. I hope it wasn't impossible to do those matching. I hope you were able to do it. And Harrison, and having that dictionary of botany was helpful. Actually, Google or Wikipedia might have been useful some too. Oh, related to books, some of you may have noticed, I don't know if you're going to the holiday party, but it came across in an email recently about that announcement. I won't be there. I've got a commitment in Naples, Florida on the 3rd of December, but they're going to be having, thanks to yours truly for, as Steve says, priming the pump, there's going to be a book auction. In fact, that announcement is already, Linda's part in has already been sent out in an email, and Steve and Ronna are going to bring the books and get rid of them. They've been weighing down their floor. I had so many books in a house in Augusta one time, I sank my oak floor 2 inches. So I had to reinforce it. So I learned about building libraries then, that you better do something before you put a bunch of books in it. But all right, so we wound up with 10 participants. I guess that includes me. But so
Charlie just joined us.
Who did?
Charlie.
Yeah, I thought I saw Charlie come in there and smiling face. But well, I'm gonna I'm recording it now. So for Charlie and anybody that didn't come at all will be able to see it. See what I said in the beginning, which was fairly useful, I think. I have one thing in common with our former President Trump, is I like to talk. So that's about all I have in common with him, I'm afraid. But anyway, so, but what I did say was fairly useful, and we're gonna, I'll make sure that recording, when it gets processed, goes out to everybody, Charlie. So, all right. All right, no further questions, we'll disband. Short, that was short. 25 minutes, that must be a record. All right. All right, bye.
Bye.
Bye. Thank you.
Welcome.
Bye.
Thanks, Bobby.
All right, you're welcome.